How do you judge human risk before a medicine reaches people? Toxicology gives that early view by testing hazards, dose relationships, study quality, and the limits of animal to human translation. This walkthrough follows the non-clinical safety logic that supports safer development and stronger pharmacovigilance decisions. Toxicology studies the adverse effects of chemicals on living systems. In drug development, it defines hazard before any person is exposed. A core rule credited to Paracelsus is that the dose makes the poison. Toxicity can reflect intended pharmacology, off-target effects, or chemically reactive metabolites. Acute toxicity follows a single or short exposure, while chronic toxicity develops over repeated dosing across longer periods. Local toxicity appears at the site of contact, such as injection site necrosis. Systemic toxicity follows absorption and distribution to distant organs. Some effects reverse after dosing stops, while others persist. Dose response is the foundation of toxicology. As dose rises, severity usually rises, and more subjects may be affected. A graded response tracks increasing severity in one individual. A quantile response tracks how many in a population show a defined effect. Threshold effects explain why a no observed adverse effect level can exist. Therapeutic index compares the dose-causing toxicity with the dose-producing benefit. A narrow index leaves little room for error. Warfarin, digoxin, lithium, and phenytoin are familiar narrow therapeutic index medicines that need monitoring and close attention to interactions. Type A reactions are dose-related, while Type B reactions are harder to predict. Before first human exposure, you generally need safety pharmacology, genotoxicity, and repeat dose toxicity in one rodent and one non-rodent species. ICH M3R2 recommends the non-clinical studies needed for each stage of clinical development and eventual marketing authorization. Pharmacokinetics and toxicokinetics are also included. The core safety pharmacology battery covers cardiovascular, central nervous, and respiratory function, and should generally be completed before human exposure. Repeat dose study duration broadly matches planned clinical duration. Up to 6 months in rodents and 9 months in non-rodents for long-term use. Developmental toxicity timing depends on trial stage. The ICHS series divides key safety questions across several guidelines. S1 covers carcinogenicity, S2-R1 covers genotoxicity, S5-R3 covers reproductive and developmental toxicity. S6-R1 addresses biotechnology-derived pharmaceuticals. S7A and S7B cover safety pharmacology, and S9 tailors expectations for anti-cancer medicines. Both begin with a bacterial gene mutation screen, commonly known as the Ames test. S5R3 looks for effects on fertility, embryo-fetal development, and prenatal and postnatal development. S6R1 recognizes that biologicals need a pharmacologically relevant species. Good laboratory practice is a quality system for how non-clinical safety studies are planned, performed, monitored, recorded, reported, and archived. The OECD principles are the recognized international standard for regulatory safety studies. GLP proves the study happened as reported, but it does not by itself guarantee strong scientific design. Each study has a named study director with overall responsibility, supported by an independent quality assurance program that inspects and audits. Standard operating procedures, defined raw data, secure archives, and fully characterized test items are mandatory elements. Pivotal toxicology studies for regulators must be GLP compliant, while exploratory work often is not. Key dose descriptors organize non-clinical interpretation. NOAEL is the highest tested dose with no significant increase in adverse effects versus control. LOAEL is the lowest dose with an observed adverse effect. Maximum tolerated dose is used mainly in oncology and carcinogenicity design. Human equivalent dose and maximum recommended starting dose support first human dosing. Human equivalent dose uses body surface area scaling rather than simple body weight scaling. In the worked example, a rat NOAEL of 50 milligrams per kilogram per day becomes 8.06 milligrams per kilogram after division by 6.2. Applying the default safety factor of 10 gives a starting point of 0.81 milligrams per kilogram. The same worked example compares a dog NOAEL of 15 milligrams per kilogram per day, which becomes 8.33 milligrams per kilogram after division by 1.8. The rat is therefore the more sensitive species. Even so, starting dose remains a judgment supported by arithmetic, not a number produced by arithmetic alone. For higher risk products, the starting dose may need to relate to the minimal anticipated biological effect level rather than NOAEL alone. MABEL is derived from pharmacodynamic data, including ex vivo and in vitro studies in human tissues where feasible. In healthy volunteers, exposure should stay below the pharmacologically active dose unless well justified. The EMA First Human Guideline, Revision 1, was adopted in July 2017 and applied from February 2018. It expects careful risk control when uncertainty is high. Sentinel dosing means one subject in a cohort is dosed first. Protocols also need clear stopping rules for serious or severe reactions within a cohort. TGN 1412 was an anti-CD28 monoclonal antibody given to 6 healthy volunteers at Northwick Park in London in March 2006. All 6 developed a severe cytokine release reaction within hours and needed intensive care support. The case showed a major failure of species translation for an agonist biological. The consequences were practical and lasting. Wider use of MABEL followed, along with sentinel dosing, slower escalation, careful site selection, and explicit stopping rules for higher-risk products. The revised EMA guideline embedded these expectations in integrated protocols for single ascending dose and multiple ascending dose parts. The 3 R's are replacement, reduction, and refinement. Replacement uses predictive models and tools instead of animals. Reduction designs robust experiments so fewer animals answer the question. Refinement minimizes pain, suffering, and distress, while recognizing that animal welfare affects scientific quality. New approaches include human hepatocytes, HERG patch clamp, and organ-on-a-chip systems. Drug-induced liver injury is a leading cause of drug withdrawal and a leading cause of acute liver failure in Western countries. HIS law helps identify serious hepatotoxic risk in pre-marketing evaluation. It needs hepatocellular injury, bilirubin above the threshold, and no better alternative explanation. Rising bilirubin usually follows aminotransferase increase by days to weeks. In the illustrative case, alanine aminotransferase reaches 6.5 times the upper limit of normal, and total bilirubin reaches 2.75 times the upper limit. Alkaline phosphatase stays below twice the upper limit. supporting a hepatocellular pattern rather than cholestatic injury. With other causes excluded, the case meets Hy's law and warrants urgent action. The kidney is vulnerable because it receives high blood flow and concentrates solutes in the tubules, with aminoglycosides and cisplatin named as classic nephrotoxins. Cardiotoxicity includes direct contractile injury and QT prolongation, which can lead to torsades de pointes. Hematological toxicity ranges from anemia to agranulocytosis, and immunotoxicity includes hypersensitivity and cytokine release syndrome. The later questions and answers describe a double negative strategy, meaning an adequate HERG safety margin plus no QTc prolongation in a well-conducted in vivo study. Even then, translation from animals to humans remains imperfect. Non-clinical findings identify hazard, but human risk depends on exposure margin, species relevance, and mechanism. Some animal findings never appear in humans, and some human reactions never appeared in animals. In pharmacovigilance, toxicology supports the risk management plan safety specification and adds biological plausibility to causality assessment alongside temporality, de-challenge, re-challenge, and alternatives. You have seen how toxicology moves from basic dose principles to study design, dose setting, first human safeguards, organ-specific toxicity, and risk translation. The main lesson is simple. Non-clinical data can guide safer decisions, but it never removes the need for judgment. That is why toxicology remains central to both development and everyday pharmacovigilance.